Western blotting is an important technique used in cell and molecular biology for protein separation and detection. It enables the separation and identification of a specific protein of interest from a complex mixture of proteins, for example a cell lysate. A typical Western blot technique includes the following steps: separation by size, transfer of protein to a solid support, and detecting target protein using antibodies. To separate the macromolecules in a sample, SDS page technique can be used. The first step in the SDS page procedure is sample preparation, so that a loading buffer containing SDS, beta-mercaptoethanol, bromophenol blue and glycerol is added to the protein samples. The loading buffer is used in order to give all proteins present a uniform negative charge, since proteins can be positively, negatively, or neutrally charged. Once the loading buffer is added, the samples are then heated to 95 degrees Celsius. Proteins are large biomolecules consisting of one or more long chains of amino acid residues. They are formed by linking amino acids with peptide bonds and fold into specific spatial conformations driven by a number of interactions such as hydrogen bonds, hydrophobic interaction, disulfide bonds, ionic bonds. SDS is an anionic surfactant that contain a polar head group with a net negative charge at the end of a long hydrophobic carbon chain. SDS denatures the native proteins by disturbing the non-covalent forces, include hydrogen bonding, hydrophobic, and ionic interactions, while the reducing agent beta-mercaptoethanol is used to cleave the disulfide bonds. Also, SDS binds fairly uniformly to the protein, and the intrinsic charges of this protein become negligible when compared to the negative charges contributed by SDS. This treatment brings the folded proteins down to linear molecules with net negative charge. Therefore, these proteins can be separated in a polyacrylamide gel based on their molecular weight. The gel is produced by polymerization between two glass plates anchored vertically in a cassette. The gel cassette is placed vertically between two electrodes. Positive electrode located at the bottom of the gel, whereas negative electrode is positioned at the top of the gel. Next, the gel is inserted into a chamber. Then a running buffer is poured to allow the conduction of current through the gel. SDS is also present in the gel and in the running buffer to make sure that once the proteins are denatured, they stay that way throughout the run. During separation, a molecular weight size marker is usually loaded onto the gel. This consists of proteins of known sizes and thereby allows the estimation of the sizes of the proteins in the actual samples, which migrate in parallel in different tracks of the gel. Each sample is added into its own well in the gel. As we have seen previously, bromophenol blue and glycerol are present in the samples. Bromophenol blue is a dye that is useful for visualizing the sample in the well, while glycerol increases the density of a sample and it is used to fall the sample into the well. After the sample application procedure, an electric field is applied across the gel, causing the negatively charged proteins to migrate across the gel, away from the negative electrode and towards the positive electrode. Also, small proteins migrate relatively easily through the mesh of the gel, while larger proteins are more likely to be retained and thereby migrate more slowly. Due to the relatively small molecule size of bromophenol blue, it migrates faster than proteins. And by optical control of the migrating colored band, the electrophoresis can be stopped before the samples have completely migrated through the gel and leave it. Once the proteins are separated, the gel cassette is removed from the electrophoresis tank. Next, the glass plates are removed, and the top portion containing the wells is cut off from the gel. In the next step, the separated proteins will be transferred from inside the gel to an appropriate membrane. When performing a wet transfer, the gel is first equilibrated in transfer buffer, so that it is placed in a tray with the blotting buffer on a rocking platform. To make a blotting sandwich, a gel holder cassette is immersed in the transfer buffer, with the black side down and the white side up and out of the buffer. Next, a fiber foam pad is immersed in the transfer buffer. Then, it is laid on the black side of the gel holder cassette. After that, a piece of a filter paper is soaked in the transfer buffer. Then, it is placed on top of the fiber pad. Once the gel has been equilibrated in the transfer buffer, it is carefully placed on the blotting paper. For the transfer, a nitrocellulose membrane or a polyvinylidene difluoride membrane can be used. The membrane is soaked in the transfer buffer. Then, it is placed squarely on the gel. Next, a second sheet of blotting paper is wetted with the transfer buffer. Then, it is placed on top of the membrane. Finally, a second fiber pad is immersed in the transfer buffer. Then, it is laid on top of the blotting paper. Next, the gel holder cassette is closed and locked with a clamp. The most commonly used method for transferring the proteins is called electroblotting. During this transfer, an electrode assembly with positive electrode and negative electrode is used. The gel holder cassette is inserted into the module, ensuring that the cassette is properly positioned from negative to positive. The inner module is placed into the electrophoresis chamber. Then, the chamber is filled with the blotting buffer. Next, a lid is placed on the electrophoresis tank, and electrical leads are connected to the power supply to run the blot. Electroblotting uses an electric current to pull the negatively charged proteins from the gel towards the positively charged electrode and into the membrane. The proteins move from within the gel onto the membrane while maintaining the organization they had within the gel. When the run is complete, the electrophoresis chamber is disassembled and the inner module is removed. Then, the gel holder cassette is removed and opened. Next, starting with the fiber pad, each layer is removed until reaching the membrane. After the electrotransfer of the proteins on the membrane, the next step is the detection of our protein of interest using antibodies. First of all, the membrane is soaked in a blocking solution, which contains bovine serum albumin. This step must be taken to prevent the interactions between the membrane and the antibody used for detection of the target protein. Once the blocking solution has been added, the membrane is incubated on a rocking platform. The BSA protein attaches to the membrane in all places where the target proteins have not attached. Thus, when the antibody is added, it cannot bind to the membrane, and therefore the only available binding site is the specific target protein. After the incubation is complete, the blocking solution is removed. Then a solution containing the primary antibody is poured into the tray. Next, a membrane is incubated under gentle agitation. The primary antibody binds to the specific target protein. Following incubation, the primary antibody solution is removed. Then a wash buffer is poured. Next, a membrane is incubated on the rocking platform. The membrane is washed several times in wash buffer to remove unbound primary antibody and thereby minimize background. After washing the membrane, the wash buffer is removed. Then, a solution containing the secondary antibody is added into the tank. Next, a membrane is incubated under gentle agitation. The secondary antibody recognizes and binds to a specific portion of the primary antibody. Following incubation, the secondary antibody solution is removed. Then, the wash buffer is poured into the tank. Next, a membrane is incubated on the rocking platform. The membrane is washed several times in wash buffer to remove unbound secondary antibodies. It is crucial to thoroughly wash the membrane at this step. After rinsing the membrane to remove unbound secondary antibody, the membrane is incubated in a solution containing the substrate. Chemiluminescent Western blot detection is the most frequently used method to detect proteins on Western blots. To allow detection of the target protein, the secondary antibody is commonly linked to a reporter enzyme such as horseradish peroxidase. The most popular Western blot substrates are luminol-based. In the presence of a hydrogen peroxide, horseradish peroxidase enzyme catalyzes the oxidation of luminol to excited state product called 3-aminophthalate. This product decays to a lower energy state by releasing photons of light at 425 nanometer. The production of luminescence is proportional to the amount of horseradish peroxidase conjugated secondary antibody, and therefore indirectly measures the presence of the target protein. After incubation, the membrane is placed in a clear plastic wrap to prevent drying. The membrane is analyzed by densitometry, where the resulting light signal is detected by CCD camera which capture a digital image of the Western blot. According to the obtained bands, we can determine the approximate size of our protein of interest, as well as its presence or absence in each sample.